This topic is all about cells, cell structures, microscopes, the basics of cells. When we're talking about cells, now someone didn't just one day wake up and was like, you know what would be funny if we were all made up of cells. It was a thing that happened in slow and like long-term discoveries. So at the beginning of the quarter, you guys should have done puzzle theory, where we gave you guys a piece of a bigger puzzle and you guys basically slowly put it together. And as we gave you more and more pieces, the pictures all just came together. This is basically how it worked for when we went over cells. So at the beginning, you have this guy, his name's Robert Hooke. Basically, he takes a sliver from a cork and he puts it under a microscope and he sees that it's made up of all these little things that he calls cells. He calls them cells because he thought they were hollow inside. Now, you guys have probably had this shoved down your throat since you were little. Cells have things inside of them. Cells are not empty. But we didn't know that at the time. Then you had this other man, Leeuwenhoek. I don't know. I'm not going to be able to pronounce that right. I'm not even going to try. All right. So basically, he was like, corks are great, but we're going to try other things. So he took samples of pond water and swabs from a human mouth, and he saw that there were these things called bacteria. So now we're slowly building up. Before you had empty things, now you have bacteria. So we're getting somewhere. Then he observed that basically cells were everywhere. Cells became the basic unit of light. And as time went by, they were like, wow, plants are made out of cells. Animals are made out of cells. And eventually you have the cell theory as it is now. Now, how do we know so much about cells? Microscopes. So you have the basic light microscope. If you guys ever used a microscope in class, odds are you used a light microscope. It's very basic. You have the lens, it sends a light through the specimen and you guys can see it. Now you would be able to see small things, like if you guys had been in lab for the plant section, like in one-on-one, not pandemic year lab, you guys would have had the opportunity to see things like plant cells, how cells divide, root tip cells. So it gets the basic outline, but you're not going to see any more than that. Now, if you wanted to see more, you're going to want to go with an electron microscope. They're basically going to just pelt it and give you a more defined image. They tend to have a higher resolution than light microscopes. You also have transmission and scanning microscopes. They're subdivided. Transmission ones make it so that you can look at the larger proteins and the cell structures. The problem is that the beam can only pass through a thin sample, so you have to make sure that it's cut super thin. Also, they're two-dimensional images that you're going to see. It's not going to be a 3D image. The scanning electron microscope is going to be a little different. So they take surface images. So you don't have to cut them into slices because it's just the surface that you're looking at. And because it is the surfaces that you're looking at, it's going to give you a three-dimensional image. I don't know why I forgot that word. I'm going to move on from that. We've gone over this already, prokaryotes and eukaryotes. You guys already know prokaryotic organisms are usually single-celled organisms. Eukaryotic organisms go through cell division, and most of the time they actually have sexual reproduction. They also are separated because in the cells, the prokaryotic ones basically just have, they're kind of like soupy inside. Eukaryotic cells don't. Everything's nice and organized, the nucleus, and you have the genetic material in the nucleus. Prokaryotic ones, like I said, are kind of soupy, so everything's just kind of floating around. So all cells have these things called a cell membrane. We're going to talk about it when we go further on into the chapter, but just know that a cell membrane is found in all the cells. However, there are some cells, like plant cells, that will have this extra bit of protection that is called the cell wall. Now, the cell wall is going to provide additional protection and it's going to give it a structure. That's why if you look at a plant, most of the time they're standing up straight and rigid. Their cells are also straight and rigid. So you have the nucleus. The nucleus is basically going to be like a lockdown area with super important information, in which case it's going to be the DNA. So you guys can see the difference. You have the prokaryotic cells and the eukaryotic cells. The ones that we're going to be dealing with the most are going to be the animal and plant cells. However, like we saw when we had, what was it? The classifications. Protists are also eukaryotic. But just so you guys can see the main differences, we're going to focus on animal and plant cells. So prokaryotic, again, they're very basic. They just respond to the environment. If they go through cell division, they're going to end up making clones. Animal and plant cells will only do that with body cells, but you guys will actually go through sexual reproduction because you are animals. Let's get through that. Now, cell organizations. You guys are going to do this in lab, or more likely do in lab. We're going to have this project called a cell city. So you guys are going to take what we're going to teach you today, and you're going to make this into an actual city. You're going to give every part of the cell a job. So we already talked about the nucleus being basically the town hall of the cell, with all the important stuff being inside, such as the DNA. So the cytoplasm is going to be on the outside, the fluid portion. Everything outside, it's more like a sidewalk or a street, except it's kind of jelly-like. So that's the basics. After that, you're going to look at the smaller parts of the cell, the organelles. The organelles are going to be like the small organs that help everything function, where we went over the nucleus. Just going to go over that. So they do have pores. Information will come through the pores out into the cell in order to help you guys make things like proteins. You also have these things called vacuoles. They're going to be in charge of storage, such as water, salts, proteins, carbohydrates, and how they get there, you'll see. The plants and the animals differ in the plants have a bigger vacuole, which will have a lot of liquids inside. All right, so the lysosomes. The lysosomes are going to be kind of like your trash can. They come around and they break things down into smaller molecules that can be used by the rest of the cell. Or if it doesn't need it anymore, it kind of just gets rid of it. Then you have the cytoskeleton. So the cytoskeleton is basically exactly what it sounds like. It's the cell's skeleton. It's going to help it keep its shape, but it's also going to make sure that it can transport things to different parts of the cells. Now, within the cytoskeleton, you have two different parts, which are the microfilaments and the microtubules. The microfilaments have actin in it, and they have a whole bunch of networks that help with the actual shape. They also help the cell move, and they allow for like a more flexible structure. Now, when we go to the microtubules, they're going to be a little different. Think of like steel rods. They're going to be hollow inside. They're going to help and maintain the shape. They are going to show up again. You will see them when we go over mitosis, that they help separate the chromosomes, but that we'll worry about later on. The centrioles, same thing. They're going to be located near the nucleus, and they help organization during the cell division, but again, we're not there yet. All right, so how do we build proteins? Now, we went over some of the basics. You guys may already know this. So you have these things called ribosomes. In the animal cell, there can be ribosomes scattered all around, and there's going to be some in the rough yard. So the ribosomes are basically in charge of making those proteins, or at least starting the process. So you're going to have your actual cells assembled. The proteins are going to start assembling the proteins are going to be assembled by the ribosome. Then they're going to slowly be exported, but now it needs to hit a certain amount of spots. We already talked about how it goes through the ER. Depending on which one it is, some of them may already have ribosomes in them. So the rough ER is going to basically the rough ER's main job is to make the transport proteins that we're going to get from these ribosomes. And the smooth one is going to make fats and membrane proteins, and it doesn't contain any ribosomes at all. So this is the whole process. They basically start assembling everything. It goes from one organelle to the other. Eventually, it's going to end up at the Golgi apparatus. If you guys have ever delivered a package or returned anything from Amazon, think of that as your packaging center or UPS. They're going to go ahead and they're going to transport the proteins wherever they need to go. You guys already know the chloroplast is basically an extra plant version of the mitochondria, where plants get to do their thing, make their own energy using sunlight. And we have the mitochondria. Guys, when we're going over these organelles, please never tell me or Giannis or any bio teacher here that the mitochondria is the powerhouse of the cell. I understand that it helps you guys understand that it makes that energy, but you guys do not understand the process it goes through. And until you do, we don't want you to call it that because it's a cop-out. All right, so when we're going over the cell walls and the cell membranes, we already talked about how they're kind of like an extra protection, but as you saw over here, it does the proteins and other things need to come in and out of the cell. So it needs a specific way to do that. So the cell membrane has this thing called a lipid bilayer. So a lipid bilayer just means that you have 2 layers of lipids. So what's a lipid? It's this thing. So it has this thing called, wait a minute. Okay, yes. Okay. So the head is hydrophilic and the tail is hydrophobic. This confused me, guys. Sorry about that. So what that means is basically that the head likes water, the tail does not. That's why you get that shape. So if you look at it, you'll see how this one's standing up over here, but these are the opposite. So basically it means if I have water on the outside, if I have water here, then the head will come this way, but the tail will go that way, which means I should have some kind of water-like thing out here, and that's why that orientation happens. Now this creates this thing called a semi-permeable membrane, which means only certain things can come through, which is fine because we have different ways of doing it. If I had you guys coming into my classroom, you guys could go through the door. I'm going to allow certain people in, certain people out. I'm not going to allow like kids that don't belong to this class to go through. That would be your semi-permeable membrane. Now, if you're trying to get into my class, but not through the door because I'm not letting you through, you could try to find maybe a window. Yes, or maybe you're going to try to go through a different room and just blast through the wall, or you're going to send a message through text. In science, everything wants to move in the way that is easiest to do so. So your cells are going to want to go from high to low. So if I'm holding something up here and I throw it so it hits the floor, gravity did all the work. I did nothing. That is the way that your cell wants to do things, but not specifically with gravity. Now, if I threw my pen just like that, took me no effort. Now, for me to bend down and actually pick that pen up is going to take effort, and that is exactly what's going to happen with active transport. Diffusion is going to be the easy one. So if I have like 7 molecules over here and 2 here, it's going to go from high to low. So it's going to go this way. But eventually I may get too many on one side and it's going to go the other way. You guys should have seen this when we went over the electron transport chain. The hydrogens kind of go in and out, and that's what helps you transport everything. So all you need to know, diffusion is for literally anything that's not water. Water itself has its own little special one, and that's called osmosis. So facilitated diffusion. I already told you guys diffusion is basically when something goes from high to low. Facilitated diffusion is exactly what it sounds like. It's going to be when you go through diffusion, but with assistance. So you're going to have this channel, this protein channel, that's going to help you get those things from high to low. And it's going to go right through the cell. Why? Because whatever permission this needs to go through the actual membrane isn't working. They don't have that kind of permission, so they got to come through another way. And that's why they have to go through facilitated diffusion. Now, again, I told you osmosis is when it goes from high to low. It's the same thing. It always goes through a semi-permeable membrane. Diffusion also, again, goes through a semi-permeable membrane. Now, I think here it is. So you're going to end up with things such as isotonic, hypertonic, and hypotonic. When you're looking at the word, just look at the word. So if I have a lot of sugar, let me see if I can get the drawing tool. All right. All right, so first, hypo, below, hyper. If I gave you guys a lot of sugar, you would be hypertonic. You would be super excited. Hypotonic would be below. So if I had a cup here, separated by a membrane, and I had a whole bunch of stuff here, and only one here, again, it's going to go from high to low. But as of right now, this one's hypertonic, this one's hypotonic. So then if this is a semi-permeable membrane, it's going to go this way. It happens the same way in your cells. So they need to find a balance. Oops. And when they find that balance, they'll end up being isotonic, which basically means everything is in balance. They'll be evened out. You'll have the same amount on one side as you will in the other one. And it doesn't mean that it's not moving. It just means that it will be moving in and out at the same rate. So it can remain in that balance. All right, so the osmotic pressure is just the force that you need in order for the osmosis to take place. Now, I told you guys that it happens in your cells, and that's exactly what happens. Now, if you have too much water coming in, it's going to burst. If you have too much water coming out, it's going to shrivel up. So in an isotonic solution, your blood is fine. In a hypertonic solution, your blood would shrivel out. In a hypotonic solution, your cell would fill with water, and at times it could just burst. It doesn't necessarily need to, but it could. Now, active transport, I told you guys, is when I'm putting energy into it. So it would be going from low to high. It's basically like a salmon swimming upstream. You don't want to do that. It's too much energy. If it comes to that, your cells actually have ways of doing it. They can go through a protein that's going to shove them in the opposite direction. Again, just like a salmon going upstream. It's a pump system that kind of shoves it into the membrane or out of the membrane. You can also go through these vesicles. They're basically like tiny cars that will escort you wherever you need to go. Your cell can also go through endocytosis or exocytosis. It just means it's going to eat it or spew it out. Endocytosis means it's going to swallow it up, again, usually in a vesicle. Exocytosis means it's going to spit it out. So from a vesicle to just get out. And that's it, guys. That's our whole lecture.